Cells are the basic building blocks of all living organisms. They are like tiny factories, constantly carrying out various functions to keep us alive. Just like any factory needs energy to operate, cells need energy to perform their tasks. This energy comes from a molecule called adenosine triphosphate, or ATP, which is produced in a specialized compartment within the cell called the mitochondria. Mitochondria are often referred to as the powerhouses of the cell because they are responsible for generating most of the cell's ATP. They're like tiny power plants, working tirelessly to provide the energy needed for everything from muscle contraction to nerve impulse transmission. Without mitochondria, our cells would not be able to function properly, and life as we know it would not be possible. Mitochondria are fascinating organelles with a complex structure that is essential for their function. Understanding how ATP is made in mitochondria requires a closer look at their unique architecture. Mitochondria have a distinctive bean-shaped structure with two membranes, an outer membrane and an inner membrane. The outer membrane acts as a barrier, separating the mitochondria from the rest of the cell. The inner membrane, on the other hand, is highly folded, forming structures called cristae. These folds increase the surface area of the inner membrane, providing more space for the proteins involved in ATP production. Between the outer and inner membranes is a region called the intermembrane space. This space plays a crucial role in the process of ATP synthesis. The innermost compartment of the mitochondria is called the matrix. It contains enzymes, ribosomes, and DNA, making mitochondria partially independent from the cell's nucleus. The presence of two membranes and distinct compartments within the mitochondria is crucial for the process of ATP production. Each compartment plays a specific role in the intricate series of reactions that ultimately lead to the synthesis of ATP. ATP is a complex molecule, but its function is simple. It stores and releases energy. Imagine ATP as a rechargeable battery. When energy is available, like when we eat food, the battery is charged. This stored energy can then be released when needed to power cellular work. The energy stored in ATP is held within the bonds between three phosphate groups. Breaking these bonds releases energy which can be used by the cell for various processes. This process is analogous to plugging a device into a charged battery to power it. Without ATP, cells would not be able to perform basic functions like muscle contraction, nerve impulse transmission, or even the synthesis of new molecules. ATP is essential for life, and its production in mitochondria is a vital process for all living organisms. The journey of ATP production begins outside the mitochondria in the cell's cytoplasm with a process called glycolysis. Glycolysis is the initial breakdown of glucose, a simple sugar that serves as the primary fuel source for most cells. During glycolysis, a molecule of glucose is broken down into two molecules of pyruvate. This process releases a small amount of ATP and NADH, another energy-carrying molecule. Think of glycolysis as the first stage of energy extraction from glucose, like striking a match to initiate a larger fire. The pyruvate molecules produced in glycolysis are then transported into the mitochondria, where they enter the next stage of ATP production, the Kreb cycle. Glycolysis is a crucial first step in the overall process of ATP synthesis, setting the stage for the more energy-intensive stages that follow. Section 5. The Kreb cycle, a cycle of energy production. Once inside the mitochondria, the pyruvate molecules from glycolysis enter the Kreb cycle, also known as the citric acid cycle. This cycle takes place in the mitochondrial matrix and is a central hub for energy production. Imagine the Kreb cycle as a spinning wheel, constantly turning and generating energy. Each turn of the wheel represents one cycle, and each cycle produces a small amount of ATP, NADH, and FAD2, another energy carrier molecule. The Kreb cycle is not only important for ATP production, but also for providing building blocks for other molecules essential for cell function. It is a central metabolic pathway that connects various metabolic processes within the cell. Section 6. The electron transport chain, a relay race for energy. The NADH and FADH2 molecules produced during glycolysis and the Krebs cycle carry high-energy electrons. These electrons are then passed along a chain of proteins embedded in the inner mitochondrial membrane called the electron transport chain. Imagine the electron transport chain as a relay race, with electrons being passed from one runner, or protein, to another. As the electrons move down the chain, they release energy which is used to pump protons, or H+, from the mitochondrial matrix to the intermembrane space. This pumping of protons creates a proton gradient across the inner mitochondrial membrane, with a higher concentration of protons in the intermembrane space than in the matrix. This gradient is like a dam holding back water, storing potential energy. Section 7. Oxidative phosphorylation, making ATP. The potential energy stored in the proton gradient is then used to drive the synthesis of ATP through a process called oxidative phosphorylation. This process involves an enzyme complex called ATP synthase, which acts like a turbine, using the flow of protons back into the matrix to generate ATP. As protons flow down their concentration gradient through ATP synthase, they cause a rotational force that drives the synthesis of ATP from ATP and phosphate. This process is analogous to water flowing through a hydroelectric dam, generating electricity. Oxidative phosphorylation is the final stage of ATP production, and it is responsible for generating the majority of ATP in most cells. The electron transport chain and oxidative phosphorylation work together to harness the energy stored in the proton gradient to produce ATP. Section 8. Putting it all together, a summary of ATP production. The production of ATP in mitochondria is a complex process that involves multiple stages, each playing a crucial role. It begins with glycolysis, where glucose is broken down into pyruvate. Pyruvate then enters the mitochondria and is further broken down in the Kreb cycle, producing a small amount of ATP and energy carriers like NADH and FAD2. The high-energy electrons from NADH and FAD2 are then passed along the electron transport chain, creating a proton gradient across the inner mitochondrial membrane. This gradient is then used by ATP synthase to generate ATP through oxidative phosphorylation. In essence, the process of ATP production in mitochondria involves a series of energy conversions, starting from the chemical energy stored in glucose and ending with the chemical energy stored in ATP. Section 9. Conclusion. The importance of ATP. ATP is essential for life, and its production in mitochondria is a vital process for all living organisms. Without ATP, cells would not be able to perform basic functions like muscle contraction, nerve impulse transmission, or even the synthesis of new molecules. Understanding how ATP is made in mitochondria is crucial for understanding the fundamental processes that keep us alive. It highlights the intricate and elegant ways in which cells have evolved to harness energy from their environment and use it to power the processes of life.